and adaptation. Yesterday we began the session with a Russian fly geneticist quote. It seems appropriate for the topic today, and it being the 200th anniversary of Charles Darwin. Now we should begin with Darwin's quote. And so what I have here, it's not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is the most adaptable to change. And I think that we have a series of talks this morning that talk about mechanisms of adaptation as well as where it takes the changes that have been brought. And so I'm very pleased to have, I think we have a really great session. And I'm very pleased to have begin with Peg Riley from the University of Massachusetts, who's going to talk about, maybe it isn't the species, maybe it's the community that adapts to change.
I think you're wishing there. It's the species. All right. So thank you very much for inviting me to speak. I came to a meeting similar to this, the GTL meeting about 4 or 5 years ago, and Dan Drell loves to get me in trouble, so he asked me if I'd come and speak about the species concept. And what always can you all hear me? Yeah? What always surprises me is, though I don't actually work in this area, I like to talk about it, and it just annoys everyone. So if you're annoyed, I've probably succeeded today. But in any event, I came about 4 or 5 years ago to the GTL meeting and gave my perspectives on how the emerging microbial genomics era, which Genomes to Life essentially created and has nourished and nurtured, was impacting our view about biological diversity. And now I'm very happy to report 5 years later that the genomics era has really come to fruition on this concept. And what I'm going to talk about is whether or not Linnaeus would turn in his grave if we apply his traditional hierarchical structure. This is not working, is it? On this side? Yeah, thanks. Okay, sorry. I'm a little dyslexic here. Okay, is that better? Yeah. So would Linnaeus turn over in his grave if we tried to apply with bacteria this hierarchical structure, of course, leading down to the binomial, the species name? And so let's start because it is the year of Darwin. 150 years ago or so, Darwin was prescient in just about every area of biology, and of course, even more so in the species concept. And here's a quote from Origin of the Species. Why, if species descended from other species by fine gradations, do we not see everywhere innumerable transition forms? Why is not all nature in confusion, instead of the species being as we see them, well defined? So whether you're a bird lover like Jonathan Eisen espoused yesterday, or for myself, a lover of herpetology and mammals, we know when we see a species. There are very few instances when we're wrong. And the question is, that concept is based on the fact that there are clusters in phenotype space that we as humans observe. Now, of course, we can't observe microbes, but do they also possess these clusters? They're part of the biological world. Do they play with different rules or not? So what is a species? Well, it's essentially a group of organisms that interbreed and produce fertile offspring. Now, this is a very old definition applied principally to some of the large fuzzy things that we see around us. Can it apply to microbes that we can't see and phenotypically characterize the same way? So we know that bacteria reproduce by binary fission, right? So here's a very famous photograph of an E. coli dividing, but can we say that they interbreed? Well, we already know the answer to that. Of course they interbreed. They just do it in funky ways that we wouldn't have predicted. So one of my favorite titles of a paper in the journal Genetics is Sex with Dead Things is Better Than No Sex at All. And that's because something like Nemophilus makes a living by sucking DNA up from the environment. And that turns out to confer an evolutionary advantage to that critter. And that title actually did make it into Genetics. Rosie Redfield was the postdoc in the lab that I was in at the time, and we all had bets on whether they'd accept it, and they did. So we all know that they suck up DNA, they transduce or they conjugate. Here's this lovely a very well-known illustration where there's E. coli creating a sex pilus and passing its plasmids back and forth. Once that DNA passes, though, of course, we have to have homologous recombination, which is simply the type of recombination that we have in those fuzzy creatures that we recognize as having species. So this DNA then gets incorporated into the host chromosome if homology works, and voila, bacterial sex. Now, first of all, we weren't sure if they could have sex, and now we're worried that they have too much of it. So for Doolittle life, until we both pass on, we'll argue about how important is horizontal gene transfer in describing the diversity of life. And here's an illustration from Gary Olson, who is sort of showing Ford's view, which is that we don't have a tree of life. We have this reticulated network of things. And he would argue, although not word for word, but essentially he would say that bacteria share a pangenome. And you can't distinguish species in that pangenome because every gene is up for grabs at every moment. They pass them quickly, they pass them well. So really, we come up with a biological, bacterial species problem. Is there enough DNA exchange between lineages to obliterate species boundaries? And here's a lovely illustration by Moreno, who was showing that when we think about bacteria, often we think of this binary fission and this clonal complex emerging. Well, here's sort of the extreme Doolittle view where everything's passing between each lineage all the time, and then of course the real world will be somewhere intermediate. Is there enough DNA exchange to obliterate these lineages that emerge that we have recognized as species? In other words, does the bacterial world really look like this? Is each of these a genome that shares in these color sort of color gradations? So one genome is connected to another through a variety of gene exchanges, so that this would be the pangenome, and maybe you'd recognize that as E. coli, maybe you'd recognize that as Bacillus, but in between there's all these other things that you don't know what to call. Or alternatively, is the whole genome more of a cluster of co-evolved gene complexes that certainly do pass information, but selection's acting on this complex and maintaining it, and that's why we see the phenotypic distribution that humans can observe when you look at biochemical traits and Bacillus smiley faces and such. So what I'd like to report is sort of a population genomics view of bacterial diversity, and much as I wish I could sequence every genome like Jonathan apparently gets to do, and we're going to talk, Eddie, I still rely on multi-locus sequence typing, and so too does most of the world outside of JGI. So what is it? Well, it's a snapshot of the genome, if you will, a small snapshot, set of 5 to 7 housekeeping genes that are chosen principally because they evolve slowly, although much faster than 16S, which if you ever want to think about species, don't use 16S. Okay? Can't do it. Won't work. Get things that evolve more quickly, but slowly enough that there's never a question of homology. They can be considered selectively neutral in most cases, and they encode products that are likely to be essential. So everybody in your group will have them. Okay, so 4 or 5 years ago, I might have shown this slide, and we see this extraordinary, robust distinction between 3 human pathogens. We have Neisseria gonorrhea, lactamica, and meningitis here. And the first response, well, there would be 2 responses from the audience at the Genomes to Life meeting. First of all, it's a human pathogen, and who cares? It's got a world that, you know, isn't really what your average bacteria experiences. And second of all, Peg, if you actually looked in deeply, you'd find all kinds of stuff happening there. So let's look in deeply. And here's Neisseria gonorrhea. And yes, it is a complicated interweaving web of genes across this genome. But that's really the point. A species must share genes, or it wouldn't be a species. So now let's step out from this view at the species level to something a little bit more distant. That's this. So as soon as we step back from this network of things being passed and homologous recombination maintaining the species gene pool, you see this wonderful distinction from the things that we recognize as its closest relatives. Now, sure, are we missing things in here? It's possible. But there are more and more taxa for which we've done more and more molecular types approaches to find that diversity, and I will argue that we're not finding it. But let me show you some of the things that have emerged since my last talk at a DOE-sponsored meeting. So here's Borrelia. Yeah, there's a pathogen there. Burkholderia made my horse sick with Lyme disease, made some of you sick with Lyme disease. But actually Borrelia is a very natural, often intracellular parasite that's hanging out in trees and all sorts of invertebrate hosts. And what you find, and this is hot off the press, that the things that we've named as species, when you apply multilocus sequence typing, fall out beautifully. Nothing in between. Well, let's look at something else. This is a soil bacterium, Burkholderia. Now, sure, there are some human pathogens. Cepacia emerged, Burkholderia cepacia emerged 15 years ago as a nasty bug for patients with cystic fibrosis. It actually survives in sterilizing solution. And that should keep you up at night if you didn't know that already. But also Malii, these are human pathogens. But if you apply multilocus sequence typing to here 4 different taxa recognized, you see these beautiful clusters. Now if I go in here and blow it up, yes, I'm going to see extraordinary exchange of information. But what's exciting is the fact that you have these long branches separating these. And you'll say, well, no, Peg, look, Malii sits right in the middle of Pseudomalii. And here's the biggest problem with the history of bacterial taxonomy. We allowed medical microbiologists to name things. We should never have done so. Malii presents the same situation that Shigella presents us. Shigella isn't Shigella, it's E. coli. If it were Shigella, it evolved hundreds of millions of times, because every time you find Shigella, it's simply an E. coli that's picked up a couple of plasmid encoded genes that we care about. Malii is similar, although some of its genes are chromosomally encoded. But you can get Malii many, many times in this Pseudomalii complex. So one problem we have when we're trying to disentangle what is a good species is, how was it named originally? Who named it? And why did they name it? A well-trained bacterial systematist would never name Shigella as a species because it shares everything with E. coli, save for 1 or 2 features that happen to make us ill. So again, I would argue Burkholderia, this is a soil bacterium, it's out there, it can exchange DNA all the time. Why do we get these clusters if there's not something underlying it? Streptococcus, again, now we begin to see the kinds of problems that bother people about naming species. Here you see much less separation, and yet you still have clusters in these various species of Streptococcus. Now, what about this? Maybe this is a gradation up to these other taxa. And again, until we fill in, oh good, well, that'll end that. So until we fill in more and more of the diversity, we won't know that answer. But so far, using molecular-based approaches that don't require cultivation, we don't find them. So what other examples? So we've looked at this snapshot from around the genome, but is that missing what's really important in this species type genome? If we look down deep, will we find things that change our view of this clustering? And so here's an example from TG's group working with Burkholderia. They used, I believe it was 2,600 protein encoding genes in that phylogeny and the usual 5 or 6 in this phylogeny, and they are precisely the same story. That's Burkholderia. That's one of those soil bacteria that's everywhere, growing on bark, not a human pathogen specifically, although there are taxa that are human pathogens. But you can see there is clustering emerging at the whole genome level as well. And I'm going to have to apologize because one of our participants here emailed this slide to me last night when he heard my interest in comparing whole genomes with MLST, but I've forgotten his first name. So I apologize. This is unpublished, hot off the press from one of our own participants of Clostridia genome-based phylogeny. And although you can't see the detail, these little clusters are very well-known Clostridial species. And if you compare this with the MLST-based 5 or 6 gene version, they're essentially the same. Now, if I misspeak, that's my fault, not his. We didn't have long to talk about this data. But essentially what he was telling me was that the MLST-based view and the whole genome-based view is telling us the same story. Now, of course, yesterday's talk on Bacillus, here's some work on Pseudomonas syringae, where you line up the members of a named species and then you do things like microarray or insertion-deletion events. You are going to find extraordinary diversity. Things are coming in and out, turning on and off within these lineages all the time. And here's one argument against publishing in journals that don't allow free access. I tried to get a slide of yesterday's speaker, Ashley's data, but the journal she published in, no fault to her, wouldn't allow me in. So what I did find was her mentoring some extraordinary undergraduates, serving as a mentor to the future of women scientists. So I grabbed that picture instead. But imagine that you had an image very much like this, only with insertion deletions, and that's what Ashley showed us yesterday. What did she find when she looked in close? The multiple whole genome comparisons of Bacillus subtilis revealed extraordinary levels of microdiversity, hundreds, if not thousands, of insertion-deletion events within these things we call subtilis, and yet the lineages remain clustered. Better yet, they clustered into things that I would never have predicted, subspecies. I mean, that for me is actually going too far, believe it or not. But one could label them subspecies or subgroups, and all were well separated from their nearest relatives in the genus. So what I'd like to propose is that we now begin to bring ecology to bear on this species concept. These insertion-deletion events abound, and that is the fuel for the evolutionary fire. We think of it as mutations, but in the bacterial world, we don't have to wait for mutations. We can just pick up whole functions. And that's going to remind us that species are not static entities. They're evolving. That's the definition of evolution, changes in gene frequencies over time. So we can't expect them to be static. And we should just, enjoy the fact that they're showing us how they do their experiments. They pick up DNA and then selection X. If it's a winner, great. If not, extinction. 99.9% of everything's been tried in evolution is extinct now. So their importance in defining a species is going to be determined by their ecological and phenotypic potential. This we can usually see, that we can't. And their impact on genetic discontinuity between strains. So if you insert enough stuff that makes you so different that you're no longer part of the gene pool, well, then you're off in running as your own lineage. Whether you become a species or not is principally the impact of drift. So by employing phylogenetic and ecological information, we're going to result in a more predictive species definition. So not just what does MLST look like, but what are all those little bits and pieces that are moving around doing for us? And perhaps the best example is from Vibrio, where Martin Pols and his colleagues at MIT have looked at marine bacteria in general, but Vibrio particularly. This is a genome-based phylogeny upon which he's mapped lifestyle and geography. And what he finds is that here are critters that are within 1 to 2% sequence divergence, things that we call species, and they might be a free-living form, and its closest relative may require an algae to live on. They're hanging out in the marine world side by side. They could easily exchange genetic information, and yet they're not. They're falling out as entities, ecologically defined entities. So their ecological adaptations contribute to their maintaining these species-specific co-evolved genomes. And not just Vibrio, but many marine bacteria are showing us the same view of the world, Prochlorococcus, etc. And even those guys that we don't name, that we haven't named yet, these are things that are out in the marine environment that we have no idea. We can't grow them. We just know that they'll light up with the universal primers. Surveys of their genetic diversity reveal clusters with less than 1% divergence among isolates, well separated from other clusters. So even the things we can't name, we're beginning to get a handle on what kinds of genetic diversity and what kinds of population structure they have. So let me turn from these specific examples to sort of thinking about the whole problem, the species problem. We have extraordinary stability on one hand. Phenotypically, if you're a Bacillus microbiologist, you know the species of Bacillus. If you work on Salmonella, you can smell it. You know you have Salmonella. You know it's not Pseudomonas. So we do all agree that we know our taxa. And now we have things like MLST and whole genome comparisons that show us some stability. But at the same time, there's stuff going on in there. So detailed whole genome comparisons reveal insertions, deletions, rearrangements. How do you bring all of that together into something that might allow us to see distinction in microbial diversity? And a group, Lan and Reeves, in 1993 produced the first really good approach to this, and they don't get recognized as much as they should. Many people use the word core genome, and they don't realize that it comes right back to Lan and Reeves. They were the first people to tackle this paradox of stability and flux. And they devised a core genome hypothesis, which posits that there's a subset of genes that all members of a species share that is their core genome. And these are things that are going to rarely transfer between close relatives. Now let me be clear. Of course the genes can transfer, they just won't survive. So selection will weed them out. So they rarely transfer so successfully between close relatives. They include the things we know of as housekeeping genes, but many other genes that we wouldn't predict. And they're present in essentially every isolate in the population. Now contrast that with what they call the auxiliary genome. These are genes that putatively serve to adapt isolates to local niches. They transfer between close relatives successfully all the time. Everybody who has their favorite taxon knows antibiotic resistance genes move like crazy. Heavy metal tolerance, certain inducers of stress responses, pathogenicity genes like Shigella within E. coli. So we can distinguish between these two broad classes. So here's one illustration of this large circle being the pan-genome of E. coli, of which most of it shares with a close relative, Salmonella enterica. Then there's 8% of the genome that all the isolates we find in E. coli share, and surprisingly exactly the same amount within Salmonella. And then there are lineage-specific things or things that are shared between two but not all the isolates. And so what Lan and Reeves were proposing is this dark circle is the core genome, and this stuff out here is the auxiliary genome. And we can distinguish those by how much divergence accumulates in them. There will be divergence between core genes here and here, between the species. There won't be as much between these and these because they move back and forth and constantly homogenize themselves. So we used a method produced by Jeff Blanchard at UMass Amherst called genome flux analysis, which is essentially an all-by-all blast that identifies putative HGT events when the best blast hit occurs outside your target. So if I blast the genome of E. coli, what is my best return for each of the ORF? If it's within E. coli, that's considered more of a core gene. If it's outside of E. coli, that's considered more of an auxiliary. And of the 5,600 ORFs that these guys share, only 260 were flagged as potential HGT events. And this is like, you know, the E. coli that from Nicole Perna's early publication, I think in '04, where she showed K12, the lab rat of the microbial world, and 015787, Jack in the Box, just around the corner, if you want to experience it. I just saw that yesterday. We saw, you know, that first publication on the genomic diversity between these close relatives. Well, they're not really close. They're 100 to 200 million years apart. And that is what you're seeing all of these lineages possess. So we did this, and it turns out that far less than, this is a very conservative test, 5% of each species pangenome appears to come from outside of the species. And if you look at each gene one after the other, this is what you'll find. The core genes in this comparison of 4 genomes in E. coli, 4 in Salmonella look like that. Auxiliary genes do not. They look like this, or some combination of this incongruent tree. So one of the questions I'm asked most often is, what is the core genome? Sorry, There is no such thing. Each species will have its own core genome, and that core is going to evolve. It's going to change over time, which is what we love about biological diversity. And just when you think you get it, you'll be wrong. So for example, one might assume that antibiotic resistance genes are part of the auxiliary genome. They're involved in local adaptation. They move easily. Some are not fixed in species, meaning some strains have them and some don't. And here we have a beta-lactamase resistance gene, and we isolated bacteria from the outback in Australia and found that they look and behave just like an auxiliary gene. This BLA-TEM gene was plasmid encoded, identified in about 7 different species of enteric bacteria, present at variable frequencies within each, not fixed in any, and the levels of nucleotide diversity are quite low for Pseudomonas site 0. Meaning that these genes are moving between each taxa and being homogenized all the time. So they're not evolving in different ways between the taxa. So resistance genes are auxiliary, right? Well, here's a very close relative, BLA-OXY. Same class of resistance gene, but it looks and behaves like a core. It's chromosomally encoded. It's identified only in Klebsiella. It's fixed in that species, and its levels of polymorphism are similar to the housekeeping genes. So you have to be very careful to generalize. And for those who think that resistance genes are more likely to be auxiliary, this is the common view. This is what's really happening with resistance genes. They're evolving within species, and only occasionally in very extreme situations, like our massive antibiotic resistance selection experiment by humans, do they actually move successfully outside of the species. Okay, so let me bring this all together and wrap up this view, this genomic-based view of bacterial species concept. Pick your favorite species concept. I happen to prefer the biological species concept, perhaps because he used to come into the lab I was in as a graduate student and remind us every week of how great it was. I happen to feel this is the most inclusive, and this species concept argues that species are defined by population genetics, groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. Well, let's just futz with the words a little and modify it for use with bacteria. Groups of strains that frequently exchange or could exchange core genes, but that are relatively restricted from such exchange with other groups. So it's not a yes or no thing. You're allowed to exchange things, but you must exchange them within your species more. That maintains the cohesion. It predicts that core genes are recombined within a species more than they're transferred between, and it allows for frequent exchange of auxiliary genes. So the core genome hypothesis is wonderful because it takes the existing phylogenetic classification system, which requires DNA-DNA hybridization or 16S ribosomal RNA, and a lot of suites of phenotypic characters. And that's just a reminder of Peg's personal pet peeve. If any of you have ever named a species based on a 16S sequence, then you should pay like a speeding fine to the Journal of Systematic Biology. We have rules, and you must follow them, and 16S is nothing for species. Okay? Just, you can't do it. So it provides a theoretical underpinning to what is really just something that's nuts and bolts. There was no theory proposed to explain how this works or why this works. So far, it supports taxonomic distinctions based on our phenotypic data, although I'm willing to believe there are a lot of taxa that won't match this. It doesn't require phenotype or culturing, which I think is the most important, because what's left for us to do? Well, sure, we can clean up the taxa we know about, but what about the 99% plus that we can't culture? So it provides a simple molecular assay of bacterial species relationships of the heretofore now uncultured. So let's apply the CGH to bacteria. Requires a population sample. Eddie? Maybe I should come on sabbatical and we should talk about hundreds of genomes per species amongst a lot of close relatives. That's where we're going to get the answer. Although, to be fair, we've already got a really nice description based on a small sample of putative core genes. The MLST approach is really working quite well. So that would just be my own personal desire. A species designation is then given to a group that shares a core genome and that's genotypically separated from other such groups. You saw them with the MLST data. But nonetheless, we can't get rid of bacterial physiologists, bacterial geneticists. You have to be able to interpret data because there's always going to be taxa that are maybe incipient species, maybe recently diverged, maybe don't conform to this hypothesis, and somehow you've got to decide how to name them. So let me end with one last slide here. I mentioned before that I tend to annoy people with this talk. I hope I'm getting less annoying as the data is revealing that these clusters are there, like it or not. And we can do something with them or we can argue about them, but I propose that we come to some sort of a consensus in the field of microbiology that the data will tell us the answer. I think the answer is very clear for those things that we can culture, but there's a lot more out there that we haven't cultured. So why bother? Why bother annoying people? Well, here are the reasons. First of all, we have no choice. We have to name things. It's the law, right? And we have to follow the law. No 16S-based species. We require names for things, and this makes sense. Think about doing a blast search. Now, I've asked actually some of my students in my molecular evolution course, I tell them they're not allowed to put the real names in. They have to just use the accession numbers. And man, do they get disappointed. Try interpreting a blast without species names. Your world just becomes really ugly and depressing. So think about the talks over the last few days. Has anybody stood up and said, well, that thing that somebody called Bacillus, which just has lots of pretty faces, you know, that thing? No, they call it Bacillus subtilis for a reason. It's Bacillus subtilis. We know it is. We see it. We recognize it. The second reason is we've hardly touched bacterial diversity. We have so much ahead of us. And wouldn't it be wonderful if we can find some way to capture evolutionary history in the names? So in that, I would argue that medical microbiologists, I'm sorry if they're in the audience, should not be allowed to name taxa in the future. And rather than simply reflect our human bias like Shigella, we try to give them names that reflect their evolutionary histories. And finally, why not try to do it right? So with that, I'll just mention who does the actual work. Michelle Lazat-Winitzky and I have published one paper on this. That's our contribution to the field. I give a lot of talks on it, but I don't actually publish in this area. But now the population genomics revolution is putting us in a position where we really can start publishing observations on the species concept. And Chris Reisen and Supan Bakal, Carla Goldstein, and my colleagues Rob Dord and Jeff Blanchard have all been involved in arguing about these concepts with me. So thank you. Any time for questions or no?
Okay. Question?
I was curious, the Human Microbiome Project comes from the understanding that humans have a large accumulation of other organisms living within them. Their total genetic content is probably similar to the human genome in size. Part of the problem with microbial systematics is that we're not actually recognizing that bacteria also are hosts to a lot of other organisms that could be considered species in their own right, and that we really have to characterize those so that we can define better what makes a species and what will differentiate species. Do you have any comments on that?
Yeah, viruses are what rock, right? That's all the diversity on this planet is viral. So forget about bacterial. How do we deal with those? Well, I can't even begin to wrap my brain around virus systematics. So I confess that at that level, I don't know how we think about viruses, and those are just one example of what you're talking about. But I think what you're getting at is that there are interactions, ecological interactions, that are going to impact how we think about classification. And as long as we step back and take a population genetics view, and what I mean by that is disentangle what is the population that you're studying, pull it away from what it's impacting, either being impacted by in terms of a virus or impacting in terms of it's an intracellular parasite in something and they're co-evolving. I'm not sure if I'm answering your question, but I think you just have to step back and take a population genetics viewpoint. The microbiome project is going to be extraordinary in terms of how we view the coevolution amongst some of these major groups that are required in our gut to be a healthy human. Sure, there's going to be coevolution, but my prediction, and Jeff Gordon will bear this out with all of his wonderful work on the microbiome, is that we're going to see beautiful lineages of E. coli hanging out on the gut eating mucin. And salmonella won't be there the way E. coli is, for example. Did that help?
That's sort of. I'm not sure if that completely answers the question. We're going to talk later.
Sure.
Hey, a question about choosing your MLST genes. How do you actually change? Is there a concept where you would actually have to choose a different set of MLST genes when you're looking at things that might not be that closely related? And secondly, would you actually be able to go down to a finer distinction, or would you actually not have a consistent description of differences if you just choose a certain set of MLSTs because it
Right.
reveals the very fine gradation
Yeah, sure.
that exists?
So I think I got your question right. So how do you choose your MLST genes? And if you chose a different set, what would the answer be? And we're just now in the position where we can do those experiments. So I don't have a well-informed answer, but what we can do is take things like the data sets Jonathan Eisen are producing, the data sets from Burkholderia, for example, where we have multiple whole genomes, and we can go in and choose, on average, any 5 genes, what's the answer? So far, and I don't think those are published, but so far they don't produce a different result with Burkholderia. Now, we're not going to know what genes to choose in things that are not culturable. So we're going to have to take the best guess from its closest relative, and again, that's what Giba is going to help us understand. Who are its closest relatives, and can we take the genes that we use in them and apply them in this unculturable? So, that's why we need bacterial physiologists, systematists. DNA sequences aren't going to solve the problem. They'll help us solve the problem, but it's still going to take a lot of thinking and large data sets and probably another 200 years before we really figure it all out. But we're certainly getting closer. So I wish I could tell you what genes to choose. Most people tend to agree in a certain area what are the best genes. With respect to your question of can you go out further and further, I focus on the species. I can't comment on the higher levels of the hierarchy because those are things that, unless you know the organism, it's going to be very hard to make predictions for me. Maybe if you know the organism, it'll be easier. But what I do know is that you're looking for genes that are recombining at the species level. So you don't want to choose things that are too divergent, because that means you're comparing things that aren't species, right? They have to be within a few percent sequence similarity. That's so far, anyways. That's what we see.
You actually choose your MLST set divided between the core genome and the non-core genome. That actually helps a bit.
I wouldn't choose to do that. If I'm interested in what the species is, then I need the core genome. The auxiliary genome is going to tell me what adaptations are passing through the species, for example. But you're not going to be able to find those auxiliary genes in every isolate. So if you want to define a species based on 100 isolates, well, 100 of those isolates better have the MLST genes. If they don't, you can't really produce comparable trees.
So the Ernst Mayr version of the species,
Yeah.
which is essentially that with which you can trade DNA, is fine, right, for bacteria. I don't think there's any laws that are broken by accepting that. But I think you're a little too narrow in your reliance on the core genome, that which is defining the genes that can be traded and recombined across the species. If you look at pretty much any large-scale sequencing that goes on, let's say 30 to 40% is the auxiliary genome. Yet any tranche of that auxiliary genome can be shared between 18 of those 20 strains, and not outside the species as defined by an LST right? So there must be limits to DNA transfer of what we call auxiliary genomes that contribute to niche adaptation and in a varying view contribute to speciation. So I guess the word caution would be, of course, you need a gold standard set to make some baseline definition with. But that concept is then going to be limited, biology to limit population genetics, right? Who are your neighbors? How many of them are you? What's the influx and outflux of the population? But I think that there are ways to use non-core genomes to understand how a species adapts, right? So the simple example is type 3 effectors in plant pathogens are almost never found among the type 3 effectors in animal pathogens. And so, even though they're in the same environments, that set of genes either doesn't get transferred or has no adaptive value, right, once it does get transferred. So it's still I think it's a little bit beyond the core, where I would say you have to think about speciation as well as
So my response to that is, I mean, of course, the auxiliary genome is involved in the process of evolution. There's no doubt about that. Otherwise, they wouldn't exist, right? All those auxiliary genes would go extinct. But the question I'm addressing is, how do you take what is there today and use it to identify lineages? And I would argue that the only way we can do that is by focusing on the things they all share. Now, that doesn't mean lose all those auxiliary genes, because those are, like Fred Cohen would argue, those are the ecotypes that may impact the entire species at some point in the future, or may have in the past. They may be receding because the antibiotics were cleaned up in that pond or something, and so you're losing them. But at a certain point, they were fixed and they were contributing to the whole thing. Because with auxiliary genes, you don't have a shared gene pool, and that's my only problem with it. But I take your caution to heart. The neat stuff about the process of speciation is in the auxiliary genome. It's just when you want to name things, that's where I've side on the side of the cores.
I got mine, so I guess I can ask. Sorry, Alice. This is very, in your, I think, to my mind, it's logical that we should be defining species based on genome comparisons like you're presenting. But what you've shown is that the homologous recombination is the cohesive force holding those species together. But as far as I understand, MLST has shown that some species just have very minimal or completely absent homologous recombination. So they're asexual. So what's holding those species together?
Give me a name of one. That would be my answer.
Anthracis.
Oh, Bacillus anthracis. Oh, for goodness sakes. Of course it recombines. There's just no genetic variation because there's been periodic selection, and it doesn't have a long enough history to have accumulated variability. If you give Paul Keim $100 million, which I think we have given him, and you ask him to sequence a lot of Bacillus anthracis, what do you find? You find SNPs. It's just at the early stages. Or maybe it's because we care about it so much, and it's a member of something bigger that we haven't figured out yet. But I have no reason to believe, nor do I think any microbial geneticist, and maybe I shouldn't say that, maybe there are some in this audience that would argue, I mean, the mechanisms of recombination are what they are, and we share those genes with mammals and microbes and protists. We don't have to invent anything new under the sun.
So there are no asexual bacterial species?
I've never seen an asexual bacterial species. That means you can't have a species if you're asexual, right? So in a sense, I'm being circular, but no, but I mean, really, do we have any examples of bacteria that can't recombine? Maybe we haven't seen them do it in the lab, but of course we haven't. We haven't seen any of these guys, save E. coli and a few Burkholderia, and maybe some Acinetobacter. where we have really good data of natural recombination rates, I mean, I don't know, I find that argument troublesome. I don't know where to put it. I don't have any examples where we have no evidence for recombination. We used to think E. coli was clonal, for example. Well, it's recombining constantly. Every gene tree you produce can be different from the next gene tree. When you do a consensus over the genome, it always separates out from its close relatives, Morganella or whatever the other Escherichia you want to choose. But I think clonality, may just be a moment in the evolution of the species. And I need to shut up, don't I?
There must be several experiments you can do to actually physically show it, like heavy label some members of the population at C13 and then go at it and see what the distribution of the heavy label is.
Hey, there's a stimulus package out there.
Okay, I think we have a last question over here.
Actually, I can't ask the question because Phil already took it.
Where is?
So, right here.
Oh, sorry.
So I'd like to only encourage, please, in your next talk, extend your statement that men microbiology should not name fungi either.
You got it.
Great, thank you very much.
Thank you. Stuff off.